Today we're going to discuss our fourth macromolecule, which will be nucleic acids. Now, I have two examples up here of nucleic acids, RNA and DNA. I'm sure we've heard of these, right? RNA and DNA throughout the years. So, just a little bit of a prequel to this. What should we know? What are our objectives? Okay, just like always, what's one of our objectives? Okay, who can think of one of our objectives with nucleic acids? An example of a nucleic acid, right there are two of them. What's another objective? We talked about polymers and monomers. What's the monomer for a nucleic acid? That's another objective. We talked about giving examples. What else? How they are used. Their function in our body. Yeah, how we use them. How we use them. So, by the time we get completed with this, you should be able to tell me the monomer of nucleic acids. You should be able to tell me a function in our body of nucleic acids. And you should also be able to tell me what they are composed of. So, nucleic acids. Chomp. Chomp. That is what they are composed of, what they contain, what elements. Carbon, hydrogen, oxygen, nitrogen, phosphorus. Okay? Chomp. The monomer for nucleic acids are something that we call nucleotides. The monomer for a nucleic acid is what we call a nucleotide. Now, what is a nucleotide? Well, it has three parts: a sugar, a base, and a phosphate group. A sugar, a base, and a phosphate group. The function of nucleic acids are to store genetic information. To store genetic information. We know the DNA of a cell is located where? Where's the DNA of a eukaryotic cell located? In the nucleus, right? In the nucleus. So, the genetic information, the nucleic acids, the DNA, is in the nucleus, and it stores the genetic information. Examples of nucleic acids are DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. We'll get more into depth over DNA and RNA when we do our central dogma. I talked about earlier in the class that, DNA makes the RNA. The RNA is then read to make the protein at the ribosomes. So, we could say that these proteins have a big, the construction of the protein comes from this nucleic acids, these nucleic acids, the DNA and the RNA. If you look right here, here is a nucleic acid. Here is a nucleic acid. And here are the three parts. The one I'm circling right here in red is the phosphate group. In green here is a sugar And in blue up here is a nitrogenous base. So we have all three parts right there, a sugar, a phosphate, and a nitrogenous base. And you can see that all of these chomps C. Okay, we can see some C's in there. We see H's, we see O's, we see nitrogens, and we also see phosphorus in the phosphate group. So all of the chomps are present in this one nucleotide. Here's another example. Here's another example that shows you the three parts. We just talked about it in green right up here. In green right here. That is the phosphate group. In blue is the nitrogenous base. And in red is the 5-carbon sugar. Now, these 5-carbon sugars, depending on what you have, okay? If you have DNA, it's deoxyribo is the sugar, okay? Deoxyribose is the sugar. If you have RNA, it's just ribose. Okay, so RNA, the sugar is ribose. DNA, it's deoxyribose. The phosphate groups are similar. The nitrogenous bases, and we'll discuss this more later, we could have different nitrogenous bases. Some of the nitrogenous bases, have you ever heard of? Okay, cytosine, guanine, okay, thiamine, adenine, uracil. Those are the 5 nitrogenous bases that go onto a DNA or an RNA molecule. But nonetheless, this is a monomer of the nucleic acid, which is what we call a nucleotide. Once again, we stated the function is genetic or hereditary information. The blueprint of life is your DNA. Now, switch over to the enzyme page. Okay, I've got two. So here is the first part of your enzymes, the activation energy. What is activation energy? It's the minimum amount of energy that is needed for reactants to form products. So in a chemical reaction, we need a certain amount of energy. We did a lab in chemistry the other day with a magnesium ribbon. Okay? We had magnesium ribbon and we reacted it to make magnesium oxide. Now, I could take magnesium ribbon and I could put it in a crucible and I could let it sit there and it would sit there for days, for weeks, for years and do nothing because I haven't hit the activation energy. However, when I would heat that, if I heat it, I get the magnesium warm enough so that it would actually react. It would pull oxygen from the air, and the activation energy would be the amount of heat. Okay, once I got it so hot, then that would react. Okay, so if I look at this diagram, okay, if I look at this diagram, right here are the reactants. Right here are the products. And in order to go from reactants to products, we need this amount of energy right here. That is what we call the activation energy. Right there. The amount of energy needed for the reactants to form the products. It's right there in your notes, isn't it? Diagram. Okay? So that is the activation energy. An enzyme. We hit a little bit about enzymes when we talked about proteins. An enzyme is a biological catalyst that lowers the activation energy needed to start the reaction. Enzymes are not used up during a chemical reaction. That's an important part. Enzymes help speed up a reaction, but they are not actually used in the reactants or products of the reaction. They are not used up. They just speed the reaction up. Now, if you look right here, here is a diagram. This is the same reaction. Okay? This is the same reaction, but look at the black line compared to the red line. See the hill of the black line. It's a lot higher than the hill of the red line, correct? Well, the red line there is with a catalyst, with an enzyme. So, you might want to jot this down right here, that this B1, that's the activation energy with the enzyme. The A1 is without the enzyme. So which one of these, okay, once you get those labeled, which one of these do you think will go quicker? Which one? With the enzyme. Why? Why is the one with the enzyme quicker? Huh? The enzyme speeds it up. See the hill? See the hill right here? If you're riding your bike on that hill, which one you get over faster? The lower one, right? So the enzyme lowers the amount of activation energy necessary, thus it speeds up the reaction. So the enzyme will lower the amount of activation energy and speed up the reaction. A substrate. A substrate is the reactant that binds to the enzyme on the active site of the enzyme. The active site forms an enzyme substrate complex which breaks bonds and makes new bonds. It'll break chemical bonds and it'll help make new chemical bonds. So, here's a diagram. I believe I have this in your book too, right? Yeah. The red part right here. This red part is the enzyme. These are substrates A and B that are coming into the active site. Right here is the active site of the enzyme. You might want to label that. You might want to label that this baby right here is the active site. That's where the substrate is going to hook up to the enzymes. Once that happens, the enzyme hooks up the two substrates, and now look, here is my new product. Has anything happened to that enzyme? No, it's free to go on to other and find other substrates and do the same thing again and again and again. Why can that happen? Because the enzyme is not used up in the reaction. It just speeds it up. It's not used up in the reaction. It just speeds up the chemical reaction. Okay? Factors. Factors that will affect an enzyme. Number one, temperature. If we heat up the enzyme, if we cool the enzyme. Number 2, the concentration. And number 3, the pH. The temperature, the concentration, the pH. Now, what we are going to be doing for a couple of days, I think it's the next 3 days, folks. The next 3 days in here, we're going to do a lab with enzymes. So we're going to see some of these things right here, okay? You're going to actually view what the temperature does. You're going to change pHs. Get a low pH, a high pH, a neutral pH, see what happens, okay? So the next 3 days, we're going to be doing some labs on, a lab on enzymes that's dealing with some of these factors, okay? Dealing with some of these factors.